So we finished in preparation for a quiz that's coming Wednesday. And the quiz material is definitely on this part, and then it'll stop. I actually have to go back and review a little bit because I'm not sure where we left off. So let's begin. This is the non-reversible enzyme regulation. And we're seeing that there is a N-terminus, and that's about all I remember about this slide. Pretty blank. We talked about non-reversible enzyme regulation. So why it's important? Well, we need it for blood clotting, right? So that is an example. And then we have other blood clotting pathologies where we have blood clots forming in the leg. We have blood clots actually forming is an embolism, the damage then floating and through the heart and getting stuck in the lungs. So that's another blood clotting issue. And then a third type of blood clotting pathologies would be in the heart, so cholesterol, and then in the brain, if we had a blood clot in the brain, in the cerebral artery, it blocks blood flow to the brain, and that is super damaging, right? So the embolus is blocking flow into the brain tissue. A blood clot breaks off from the plaque buildup in the carotid arteries of the neck, and then it plugs up in the head. And so we talked about those things. Then we moved on, and she let us know that the final exam schedule is out, so we can check that out. I will be asked to recognize some groups, which we'll show later. And then we want to talk about the cascade. So what is the cascade? Well, the intrinsic pathway, right, as this goes, I forget the numbers, but this goes to this. Is that 7? This is 10, 9, 8. 10, 9, 8, 7. Yeah. So 7 leads to 8. 8 leads to 9. 9 leads to 10. I don't remember this really well. What I do remember is that calcium is a cofactor of several proteases of blood clotting. It's absolutely necessary. EDTA can prevent that blood clotting because it binds to the calcium. How is it bound? Well, that's a good question we should ask. And then we have fibrin, which is a fibrous protein. It is not an enzyme. So there's a couple pathways there. Here's a pathway. Yeah, you definitely are going to want to review that because I don't remember a whole lot about it. In addition to blood clotting, we talked about glutamate carboxylase reaction, vitamin K antagonists. So what would an antagonist be? Well, leafy greens provides us with vitamin K. And we also get a vast majority of vitamin K from our gut flora. She didn't mention that, but on the 8th day, babies have a huge spike in their vitamin K, which is why they've recognized that on the 8th day they would circumcise a male child because they had a lot of vitamin K and they would clot up properly. Now, we had an image where we talked about ionic bonding, needing 2 negatively charged groups to bind a divalent cation process. It's called chelation. This I don't know much about. I'm going to need to ask questions. Vitamin K. So here's some vitamin K antagonists. Right, we have dicomerol and warfarin. Warfarin is actually rat poison. It was developed to kill rats, basically clot them up and cause them to die. And now it's something that's used in Coumadin, which is a statin that's to help regulate blood thickness, I believe. So why does it work? Well, because they're similar chemical structures, and they're competitive inhibitors of vitamin K at the binding site. So if we want to go back into reversible, we'll have to go back and listen at that 334 mark. That's from my notes specifically because I took them and I recorded right up there. But we're not doing that now. So we're going to switch gears and go to carbohydrates. Just review carbohydrates. So we will begin. The functions of carbohydrate, well, it's to provide energy, communication, the glycocalyx or the oligosaccharides, also extracellular matrix. It helps to organize, and we know that even it provides support structure like in the gelatin and the cow bone. And then carbohydrates also help with fiber in that all plants have the cellulose or cell wall material, right? That's carbohydrate as well. Structures of carbohydrates, they're monosaccharides. That was added in, it's important. We have the polyhydroxyl, which is an aldehyde or a ketone. An aldehyde or a ketone, they may contain nitrogen, phosphorus, or sulfur. Monomer is a building block, so that's the monosaccharide. And then we have polymers where we have disaccharides. It's two molecules that are covalently linked. This may be important. And then we have trisaccharides, which are like found in molasses. And we find out that the larger the molecule, the less sweet tasting it is. Okay, so oligosaccharides are smaller. They work more for cell-to-cell communication. They're bound to proteins or lipids and some soluble proteins. I didn't understand that statement. Polysaccharides, they range in size. Some are really small, some are really large, but they're all called polysaccharides because they're just more than 3, I suppose. And we see those as useful in nutritional energy storage. We have the cell wall, and we're kind of repeating that extracellular matrix. Now we move on to mono and disaccharides. So we have an example of an aldehyde here and a ketone here. An aldehyde has the extra hydrogen bonded onto it, and the ketone, man, they look exactly the same. I guess it's just dependent on where they are. This is a good question. Both are carbonyls. One's an aldose and one's a ketose. The ose means it's a carbohydrate. Honestly, that's about all I can pull out of that slide. The number of carbon atoms. So we want to make note that these are all found in nature, but the most important are pentose and hexose. That's 5 pairs. And she told us specifically that 4.6 billion pairs per cell. Can you imagine that? God put 4.6 billion pairs per cell. Oh, man, that's so awesome. Okay, so then we talked about sugars. We have the D and the L sugars, the D and the L sugars. So the D carbs are what's found in nature. So it's the opposite of the proteins, the aminos. Okay, so here in the Fisher projection, we're looking at glyceraldehyde, the D form, and the L form. And you can see what's switching is the OH right from left to right. They're both at the chiral group, and these two together are enantiomers, meaning they're mirror images of each other. And they have 4 different substituents. They also are optically active, so it shows up when you shine light on them. So let's move on. Then we have the D and the L sugars. So these are aldopterosis. I don't know. I'm saying that wrong. Listen to her notes. So we labeled these A, B, and C, and D so that we could tell the difference between them. We want to look for the alderosis sites by looking at the carbon furthest from something. It's like the bottom carbon in the Fischer projection. And so we know that the D-aldehydes are the ones at the bottom, but if they're not at the bottom, then it's an L, the unnatural form. And there's 4 types. We have enantiomers, which are reflected pairs. So we have B and D looking like a reflected pair, and we have A and C looking like a reflected pair. Then when they are not reflected, they're diastomers, basically any other pairings. So D sugars are determined by the configuration at the chiral carbon with the highest number. So that's, I guess that answers the question, at the highest number. Okay. So diastomers, that they only differ in one configuration. We have aldoses. So we have glyceraldehyde, ribose, glucose, mannose, and galactose. You need to memorize these. So make some flashcards out of these. And then we have ketose, deketose, that we need to know. The dehydroxyacetone, and fructose. We need to know those 2. And that was the end of lecture. And we have a quiz on Wednesday. And I am